In this lecture, we're going to try to make sense of homeostasis and how the human body maintains homeostasis even when the external conditions, either external to the cell or external to the entire body, even when these external conditions are fluctuating very wildly. So let's get started. So homeostasis is all about the ability of the body to maintain a relatively stable internal environment, even if the external environment is fluctuating wildly, because that's what the external environment does. It does not stay constant. It moves around a lot. But all the processes that are happening inside the cell must have relatively stable conditions in order for them to function at peak efficiency. So if we break down that term homeostasis, homeo means same, and stasis means standing still. So it's about keeping the internal environment within a reasonable tolerance around some value, some set point value. So homeostasis is used, these processes of homeostasis are used to maintain body temperature, the pH of our body fluids, the levels of nutrients in the blood, the levels of hormones in the blood, how rapidly we are breathing and how rapidly our heart is beating and how strong, with what kind of force the heart is contracting, what our blood pressure is, and we could make the list for days about all of the factors that are maintained in the body via homeostasis processes. So if we think about homeostatic mechanisms, then we can kind of see some general rules about how homeostatic mechanisms are maintained. So first, let's get some definitions out there. If we talk about something that is a variable, it is one of the internal conditions around which we are seeking to maintain homeostasis, and it's changeable, and it can be measured in some way by processes in the body. And for every variable that we maintain in homeostasis, there is a set point. That is, there is a normal value for this variable. So we want to keep blood sugar at a normal value, a set point. But the way that homeostasis works is that around that value, so if let's think about body temperature, for instance, 98.6 degrees Fahrenheit is average body temperature. So here we are at 98.6. But there's a range around that temperature. Anywhere between 98 degrees and 99 degrees is considered normal. And so that's well within the tolerances that the body can manage without being diseased, without being impaired or outside of homeostasis. So just because there's a set point, an average value, doesn't mean that we are always going to be at that average value. There can be fluctuation around that set point, but there are tolerances that the body can manage and that we would still consider within homeostasis. So the mechanisms of homeostasis share three components. For all homeostatic mechanisms, there is a receptor, and the receptor is what is busy measuring the variable. It is constantly tracking the variable, noting whether it is near the set point or farther away from the set point. So it notices the deviation away from the set point for the variable. There is a control center. So this is a specialized cell or a group of cells. It may be the receptor itself, or it could be a different cell that establishes what the set point is, and it determines the appropriate response when the body or the cell moves outside of homeostasis. And then finally, there is an effector. This is the tissue or organ, or this is what it is that is responding, that brings the body back into homeostasis, that brings that variable back within the tolerances around that set point. And so it's the effector that causes the effect, hence the name effector. All right, so with that definition in mind, let's take a look at let's think about some examples. And we're going to now look at how negative feedback works. So what we know is that body conditions naturally move away from the set point that is established by the control center. So for instance, it's a hot summer day, we're out mowing the lawn. Well, our body is going to get warmer than 98.6 or 37 degrees Celsius. So 98.6 Fahrenheit. Sorry about that. 98.6 equals, in Fahrenheit, equals 37 degrees Celsius. So if we're out mowing on a hot day, we're going to get warmer than 98.6. We're going to get away from homeostasis. If we decide to take a pajama walk without any jacket or gloves or even shoes on a cold, snowy winter night, our body will become cooler than the set point. If we take a brisk walk up a steep hill, we're going to see oxygen in the blood decrease and CO2 in the blood increase beyond the set points that are normal for the amount of oxygen and carbon dioxide that we're going to have in the blood. So all of this is very natural. We are constantly getting outside of homeostasis, and then the body is putting something, making something happen that will bring us back into homeostasis. So we have receptors that monitor these variables and relay information about what's happening in the body to the control centers. The control center will recognize, body is moving away from the set point, and it is going outside of the range of tolerance that we can manage. And so the control center then will make a decision about what to do, and it will activate an effector tissue or cell. And as the cell or tissue changes its activity in response to the message from the control center, then the effector tissue or cell will restore homeostasis in the body. And we call this process of, if let's think body temperature here. So if 98.6 here is the set point, here's 99, here's 98 below it. So if it's that hot summer day and we go out and mow the yard, we find ourselves getting out of homeostasis. Negative feedback, trying to figure out, there we are, it's hard to pick up that point. Negative feedback will turn that situation around, bend the curve, so we get back toward homeostasis. If we're out for our pajama walk and we get cooler than homeostasis would like, negative feedback once again bends the curve back toward the set point. So it doesn't matter whether we're cooling the body down or warming the body up. If we are bending the curve back toward the set point, then that's negative feedback. I almost think of it as a police officer standing out there with his arm, putting his palm straight into your face, saying, stop, turn around, go back. You're getting out of homeostasis. Turn around and go back toward the set point. So whether it's that we're getting too cold or we're getting too hot, we're getting away from the set point, and we have to turn that around and get back toward homeostasis. All right, so now in your notes, I've given you the home heating thermostat example. So the book uses this as a non-biological way in which, because I think we all have a kind of sense about this. So let's say we have, it's a hot summer day, we're in our house, and we decide that we want to set the temperature in the house to 72 degrees. So here we are at 72 degrees. We set our thermostat, which is the control center. We set our thermostat so that it is sitting at 72 degrees Fahrenheit, which is the set point. So if I said we're going to be on a hot day, I'm looking at my notes again and I realize, I decided that we're going to be on a cold day. So it's 72 degrees in the house if the thermometer in the thermostat notices, temperature is dropping, it can drop a little bit before the thermostat notices it. In fact, we have built into our thermostats about a 2-degree tolerance on either side of the set point. So we don't start warming the house back up again until the temperature in the house gets down to 70 degrees. When the temperature gets down to 72 degrees, the thermometer in the thermostat notices that, and it's been sending a message out back, from the thermometer to the thermostat portion that is keeping the set point. And it says the control center, this thermostat, realizes, we have now gotten outside of the boundaries of what is comfortable in the house. And so the heater will kick on, we start burning some gas or electricity, and very soon the house begins to warm up again. So when the house gets to 72 degrees, what do you think? Will that be where the heater turns off? Well, no, it's not, because it turns out that there is some variation on the upper side of this as well. In fact, the house may warm up to 74 degrees. And at that point, the thermometer will alert the control center, the thermostat, to the fact that, we've now hit 74 degrees. We're about to get warmer than we feel comfortable in our house at which point the heater turns off. Now, because it's a cool, wintry day, once the heater goes off, the house is going to begin cooling down again. And it cools down, and eventually it reaches the lower limits of the tolerance again. And once more, the heater kicks on, burning gas, warming the house up again. So we have this cycle over the course of the day where it's rare that the temperature in the house is exactly 72 degrees. So that doesn't happen very often. But at all times, the temperature in the house is maintained somewhere between 70 degrees and 74 degrees, a range in which we feel comfortable in our house. So this is how negative feedback works. As the house got too cold, the heater kicked on, and the house warmed up again. Even though the temperature is climbing, what we're doing is reversing this trend of the house getting cold. And so we hear the policeman puts up his hand, says, "Stop you. Stop getting colder," and it turns on the heater so that the house warms up. And then eventually it becomes too warm in the house, and once again the hand of the policeman goes up in the face of the house and says, "Stop. Quit getting hotter," and it turns off the heater, and then the house cools down just because houses lose heat until it gets so cold that once again the heater turns back on. Negative feedback is occurring at this moment and at this moment. So regardless of the direction, as long as it's headed back toward homeostasis, then it is negative feedback. Negative feedback is a tricky concept to get, so think about this some more. Visualize this example several times until you get it. And you could do the same thing if you are thinking about air conditioning in the summertime. It's just happening the opposite direction. So the questions then are, what is the receptor in this example? Think for a second. You can even turn off your video. Okay, hopefully you're back with me now. And the receptor is the thermometer. It's noticing what the temperature is. What is the control center? I think I've said this several times now. The control center is the thermostat, the whole box. And what is the effector? Well, in this case, it is the heater that's turning on and off, that is heating up the house, and then the outside temperature is cooling back down again, and then we heat up the house once more. So that is the cheesy non-biological example. Let's take a much more interesting example then that is biological. So as if you are out mowing on that hot summer day, and if the body temperature is increasing, as blood warmer than 98.6, and in fact warmer than 99, starts to pass through the hypothalamus, This is a portion of the brain where we have cells that are capable of responding to this increase in temperature. These thermoreceptor cells in the hypothalamus notice, oh, we're starting to warm up here, and it relays information about blood temperature to the control center cells found there also in the hypothalamus. So these are both the receptor and the control center are found in the very same place. And if you're trying to visualize where your hypothalamus is, if you imagine sort of directly behind your nose and kind of just a little bit to the front side of your ears, if you could put your fingers in those places, and then where they would intersect would be about where the hypothalamus is. So about the nose level and just in front of your ear, where those two lines intersect. That's the hypothalamus. So as the control center cells recognize that the temperature is increasing, then they send out a message, and they send it to a couple of locations. The first place is to the sweat glands, and the sweat glands, in response to this nervous, neuron firing, sending them the message, they begin to secrete a very watery sweat. And then the control center cells also send a message to the capillaries that are found near the surface of the skin. And they tell these skin capillaries to dilate, to become larger in diameter. And so this begins to shunt blood from the very warm core of the body up to the periphery of the body, right there at the skin. And it allows this warm blood to come up and circulate near the skin surface. So with all this warmth that we're dragging out of the core of the body, and all this wet sweat on the surface of the skin, we see a lot of evaporation occurring. And evaporation is a process that takes away a whole lot of heat very, very rapidly. And so thanks to all this evaporation of all the sweat that we've poured out on the surface of the skin, the body will begin to lose heat and become cooler. Now, this is harder when the relative humidity is really high outside and we don't get a lot of evaporation. Then we just get lots and lots of sweat accumulating at the surface of the body. But on a drier, warm but dry day, we can get a lot of cooling because of all the evaporation of the sweat that we're sending to the surface of the body. So, the questions again in your notes. I'm sorry, let's draw a picture here. So if 98.6 is our set point, and we're going to assume that 98 is kind of the lower tolerance and 99 is the upper tolerance. So these are the boundaries. Here we are, we're cruising along at 98.6. We get all changed into our clothes for going outside on this hot summer day and begin mowing the yard, and whoo-hoo-hoo, boy do we ever heat up. At the point where we exceed tolerance here, then the control center says, whoop, we've gotten too warm, and it sends a message out to the capillaries and to the sweat glands, and we see negative feedback. And we bend the curve back toward our homeostasis line. Now let's say that, you realized you're just about out of gas, and so you go off to the gas station, you get yourself a gallon of gas, and then you decide to go inside to the icy cold service station interior to buy yourself soda, and you go inside, and you've got all this sweat on your body, and it's cool and dry inside that store, and you're trying to find your soda, and oh my gosh, suddenly you begin to get really cold. That's because all of that evaporating sweat in that cool, dry environment is dragging your body temperature down. So in response, what you might do is get kind of goosebumpily. That is a not very useful response to getting cold for humans, because if we had more hair on us, then causing all the hairs to stand up would help to trap a layer of warm air up close to the body and keep us warm. But since we're mostly hairless animals, that's not so useful. But if we got colder and colder and colder, we would eventually start shivering. And, we've all seen this when little kids get out of the bathtub and they're all wet, and the bath has gotten kind of cool while they were in there, and they get out, and now they're out in the coolish air of the house, and they're all wet, and the water's drying off of them, and they just start shivering and shivering without, just uncontrollably. All that shivering releases heat, produces waste heat. The processes of making our muscles work produces heat in the body. And that's the reason that people who go running or exercise get all hot and sweaty as they do that. So that process of shivering hopefully will help to warm up our poor little kid's body, and it becomes warmer and gets closer again to the set point. So here at that point where we started putting up our standing our hairs on end, getting goosebumps, and then doing some shivering, that was when we began to implement homeostatic control through negative feedback. Negative, not because the temperature is rising, but because we're bending the curve. We were getting cooler, and now we're bending the curve back up toward getting warmer again and getting closer to the set point. All right, so what are the effectors in this high body temperature example? So when you were out and mowing the yard. So go ahead and pause your video while you think about that, answer the question. Okay, and I'll be back in just a second. Okay, so hopefully you thought about it for a minute. Let's see if you got it right. The effectors, when our body temperature is very warm, are the sweat glands and blood vessels. So sweat glands that are dumping out sweat, and the blood vessels, where those blood capillaries that are dilating up near the surface of the body, bringing the heat from the core up to the surface where it can be radiated away or evaporated away. So as I mentioned, I got ahead of myself, sorry about this. If body temperature is decreasing and the blood gets cooler, so this is now our pajama walk example, well, once again, we have thermoreceptor cells in the hypothalamus that relay information saying, oh, the blood's getting cooler. And in response to this recognition of cooler blood coming to the hypothalamus, the control center cells there in the hypothalamus are going to send a message out to the capillaries that are up near the surface of the skin. But this time, rather than telling them to dilate, they're going to tell those capillaries to constrict. And as they constrict, this is going to force the blood deeper into the body, trying to conserve whatever heat there is in the blood by sending it as much as possible deep into the core of the body. So keeping that blood there in the core of the body helps to prevent the loss of heat at the surface of the skin. That's the point of all that. And if we can maintain some heat inside the body rather than losing it at the skin surface, then as we do work, as the liver produces heat, as all the active organs of the body produce heat, then body temperature will rise again. So, for example, here we are once again, 98.6, here's 98, here's 99. So if we go out for a pajama walk, we start to get cold. Hypothalamus recognizes that. It institutes negative feedback here, constricting blood vessels, maybe even bringing on some shivering and bringing body temperature back up again. But it's negative feedback because we were getting cooler, and then at the point when we start doing something, telling our effector tissues to do something different, we bend the curve back toward our set point, toward homeostasis. So, in this example of the low body temperature, what is or are the effector tissues or cells in this example? So think about it for a minute. Go ahead and shut off the video. Think, see if you can come up with the answer, and I'll be back in a minute. Okay, so you're back. How did it go? Did you decide that it's the blood vessels that are responsible for helping to bring the body up? And, I said only, but I mentioned in the example as we were describing it that if we get cold enough, shivering may also happen. So the shivering muscles, if you said them as well, then you would be accurate, because I had added that in to the example as I was talking it out. Okay, so there we go. Homeostasis using negative feedback. Now, is negative feedback the only kind of feedback mechanism we have in the body? And the answer is no. There is also something called positive feedback. With positive feedback, we are reinforcing the stimulus. So rather than saying, stop, turn around, go back, we're saying, okay, you're doing something that needs to be done, do it more, and do it even more, and do it even more. And generally, positive feedback temporarily moves the body away from the normal state to get something done. And what we see is that positive feedback generally results in somewhat unstable conditions. It is, because of all the positive feedback that's happening, we're getting instability for a brief time, but it's instability that needs to happen. And so there's a couple of examples that I can give you. One is blood clotting. So if we injure a blood vessel, then we go through this multi-step series of enzymes. So we convert a material in the blood into an enzyme that will, let's say we have material A in the blood. So when we injure a blood vessel, we see the conversion of A into enzyme A. Enzyme A will help make the reaction happen so that B turns into enzyme B. So with A around, this happens more rapidly. Now, the reason that this is positive feedback is that enzyme A doesn't get used up in this reaction that turns B into enzyme B. It helps that reaction happen, and then it can go around and help another one of these turning B into enzyme B reactions to happen. So we may make just a little bit of enzyme A, but because enzyme A can do the job several times in a short period of time, we are going to get a lot more. So we get some enzyme A, we get more enzyme B, and then eventually enzyme B is going to be used to help take this material C and turn it into enzyme C. So enzyme B does that. I'm going to put EB here. And now we had more enzyme B. Now we're going to have a huge amount of enzyme C. So we go through this, we think of it as kind of a chemical cascade, where the product of one chemical reaction helps to make the second chemical reaction go faster. So we get even more enzyme B, and enzyme B helps to make the C to enzyme C reaction go even faster. And it's this multi-step. There may be as many as 7 steps, depending on which pathway we go through, that get us to the point where we produce something called thrombin. Thrombin is an enzyme that will turn fibrinogen, this is a dissolved protein in the blood, fibrinogen, into an insoluble strandy form of protein called fibrin. So fibrinogen does, or thrombin will do this. It'll help make that reaction happen. And once we get fibrin being produced at the site of the injury, that creates this big old mesh of protein that helps to capture the cells that are in the blood. Captures those blood cells, and it keeps them from, it helps to form a clot. So I'm looking in the book right now. I'm going to put you to a page. So if you've got your book handy, open it up, and I want you to look at page, almost there. Come on, really? I know there's a picture in here. Oh, here it is. On page 543 of the latest edition of the book, it's figure 1418. The caption says, "A scanning electron micrograph of red blood cells caught in a meshwork of fibrin threads." So figure 14.18 on page 543 is what you're looking at. And it's falsely colored. These scanning electron microscopes, they produce black and white images, but they colored the fibrin threads yellow, and they colored, after they took this picture, they colored the red blood cells red so that you can see this big old knotted mass of stuff that gets caught up there in a blood clot. And all of this happens very rapidly because, and we go from bleeding to clotted super rapidly because it only happens when we produce a whole bunch of thrombin, which is one of the later products. So we go through 7 or so more steps, even through some of the pathways, to get to the point where we can take thrombin and use it to convert fibrinogen into fibrin. So this is positive feedback because with each step in the process, we're getting more and more and more. We're increasing the signal that's going to be available so that we get an overwhelming amount of thrombin produced to rapidly cause clotting to happen. A second positive feedback mechanism occurs with the labor contractions. So if we've got a little baby, oh, this is going to be terrible. You're all going to laugh at me and say, what the heck? But here's a baby. It's hanging out upside down inside mama's uterus. Okay, that's a terrible baby. Let's give him a second leg. Sorry, little kid. Here's a second arm. Okay, not much better. Hopefully you get it. Okay, so here's the nose, and there's the head. Okay, so baby is inside the uterus. Here's the uterus, narrows right down to the cervix. Yeah, I get it. I am a terrible artist. So here's the cervix. Now, if, let's say it's time for the baby to be born, and at this point, at the point where we say, okay, baby, it's time to come out, the pituitary gland produces a whole bunch of oxytocin. This is a hormone that stimulates childbirth to begin. And so under the influence of oxytocin, the uterus will begin to contract. And it starts contractions up here at the top of the uterus, the fundus of the uterus. And those contractions pass like a wave down the uterine walls, forcing little baby to bump his head here against the cervix. And each time a contraction bumps the baby's head against the cervix, that sends a message. The cervix sends a message back up here to the top of the uterus saying, okay, baby's still in here. Give us another contraction. So as each contraction happens, it forces a new contraction to occur over and over and over again until finally the cervix relaxes enough and the baby winds up, is bumped into position enough so that the baby's head begins to come out through the vaginal canal, and eventually the whole body slips out or rips its way out. Don't even want to think about that, so sorry to even use those words, I apologize. And out comes the baby. And at that point, well, there's still the placenta. The placenta may flop down there and stimulate weaker contractions, but eventually the placenta gets passed as well through the vaginal canal, and once there's nothing pressing against the cervix, then those contractions very rapidly stop happening. So labor contractions are another example of positive feedback, because rather than one labor, if it were negative feedback, labor contractions would be stopped, because labor contractions are not part of the normal activity of the body, and bumping the baby's head against the cervix would just stop labor contractions. But instead, we get positive feedback, so that one labor contraction stimulates the next labor contraction to occur over and over again until baby is delivered. And it's only when baby is delivered that the contractions will then stop. Okay, so that's positive feedback, where we reinforce the stimulus, where one contraction causes another contraction to go along, or where one chemical reaction leads to the production of a little bit, the second chemical reaction produces even more, the third produces even more, until we produce an overwhelming amount of the enzyme thrombin. So there you have it, homeostasis, the attempts of the body to maintain a reasonably stable internal environment, either inside the body or inside an individual cell, so that all of the chemical reactions and other processes that are occurring inside the body or inside the cell can continue at an appropriate speed and pace. We do it primarily through negative feedback. There are also positive feedback mechanisms. Negative feedback reverses the conditions so that we restore homeostasis in the body. Positive feedback, on the other hand, is all about reinforcing, rather, pushing that same condition to happen again and again and again, so that we can resolve. Generally, it's about resolving an emergency situation, a baby that needs to come out, or a cut that's occurred in the body. All right, so there we are. Again, as always, if you have any questions, do not hesitate to ask me, and you can do that by email or by phone call or during office hours. Talk to you soon.